children, good morning everybody. In today's class we are going to have plus 2 botany chapter 3 chromosomal basis of inheritance, in which we are going to learn about mutation. Children, let me have the class mutation. What is the mutation? It is nothing but the sudden change in the genetic material of an organism is referred as mutation. What is a mutation? It is a change, just a sudden change. It happened by physical or chemical means. Okay, it was discovered by Hugo de Vries in the year 1909. Hugo de Vries. Okay, children, in the plant name evening primrose. Evening primrose is also called as Enothera lamarckiana. Enothera lamarckiana. Children, Hugo de Vries coined this and detailedly discussed in it and proposed it as what? Mutational theory. Mutational theory. Let me have in detail. Okay, children, mutation is generally classified into two types. What are they? Point mutation or gene mutation, chromosomal mutation. Two types. Point mutation is also called as gene mutation, and the second type is chromosomal mutation. What is point mutation? See, the mutational event that take place within the gene. So what are the gene protein? It is occur in the linear order in the chromosome. Is it not? When it occur any change in that, we can call them as what? A point or a gene mutation. What is chromosomal mutation? The chromosomal mutation in which the mutation take place in the number and structure of the chromosome. See, as we know, it's a thread-like structure. It's a physical basis of heredity, and where any occurrence, any change that occur in the structure and number, it is referred as what? Chromosomal mutation. Let me see agents. See, what are agents? The agents which are responsible for the mutation are referred as mutagen. The agents which is responsible for the mutation, see, which is able to occur. That is, see, when the mutant organism exposure to some mutagen, we can call them as what? A mutagenesis. The organism which is going to have that mutation is referred as mutagenized organism. Let me have the types of mutation in detail, children. See, there are about two important types. See, the mutation may occur in the single base or a base pair of a DNA. The mutation, the change that occur in a single base or the base pair of a DNA, let me have that in detail. See, what is base pair? So, for which we have to know about the detailed structure of a DNA. So, the DNA is a hereditary material which is present in the nucleus. Is it not, children? It composed of many number of nucleotides. Here I have drawn a nucleotide. What is a nucleotide? See, a sugar composed with a nitrogenous base and a phosphate, togetherly called as what? Nucleotide. What is a nucleotide? A sugar, a phosphate and a nitrogenous base. Here a doubt you will be having. What is a nitrogenous base? What are the nitrogenous bases present in the DNA? Adenine, guanine, cytosine and thymine. Children, the adenine and guanine togetherly called as purine. The thymine and the cytosine togetherly called as pyrimidine. See, these are the nucleotides. Any change occur in between the base pair, we can call them as what? A point or a gene mutation. And a number of copies in a small nucleotide sequence, if the mutation occur, that's the second type. See, children, we will have point mutation. So, what is a point mutation? The point mutation is nothing but the mutation occur within the gene. See, it is of two types. The point mutation generally classified into two different types. What are they? Base pair substitution, base pair insertion or deletion. Base pair substitution, or it is also called as what? A base pair substitution. So, one of the nucleotide will be replaced by another. The second type, base pair insertion or deletion, where the base pair may be deleted or inserted into it. See, it may produce some other protein. See, we can call them as what? Indel mutation. Indel. Insertion first 2 letters, deletion first 3 letters will be included and they will be considered as a what? Indel mutation. Let me see it in detail. Children, as I said there, the point mutation is of 2 types. What are they? Base pair substitution, another one base pair insertion or deletion. 2 types, okay? Base pair substitution and base pair insertion or deletion. That's also called as what? Indel mutation. Okay, children. So base pair substitution again divided into 2 types. What are they? Transition and transversion. Transition and transversion. Children, so the base pair substitution as well as the base pair indel mutation, that is, both make the DNA to alter. That the DNA would be what? Difficult in translating the messages which are going to encode the protein synthesis. Is it not, children? As you know, the DNA is like, the DNA is a hereditary material for every organism, and as we know, it is a double stranded, and of course it is made up of many number of nucleotides. Is it not? Each nucleotide has a sequential order, you know, it becomes triplet codon and code for a protein. These are the occurrence which will be followed, the DNA next to mRNA, next to ribosomes and all. They will be togetherly what it does? Protein synthesis. Okay, children? Those protein synthesis will be get confused by this translation. Okay, if there is a mutation in this, definitely the reading messages by the mRNA will be difficult to recall, thus the translation would be difficult. And of course, on the basis of this translation, the mutation are of 4 different types. What are they? Silent mutation or synonymous mutation, second missense mutation or esonymous mutation, the third one nonsense mutation, and the fourth one frame shift mutation. Okay, children, there are about 4 different types of mutation that came to occur after the translation. I told already, is it not? DNA is responsible for the synthesis of the protein, is it not? Which is like a chip to children, which is an hereditary material, which are more messages are encoded into the DNA, which are going to transcribed with the help of the mRNA, and later it is transported to ribosomes. It is not, then the protein will be synthesized. That will be difficult. That is difficult. After the translation is mutated, what happened? There are about 4 different mutation will be able to occur. What are they? Silent, missense, nonsense, frame shift. Silent, missense, nonsense, frame shift. So let me have it in detail. Children, the silent mutation, what it mean? One codon for one amino acid, that's the condition. Okay, so one codon, one triplet codon will be enabled to synthesize what? One amino acid later on to protein. It will be, see, since another codon of the same amino acid is encoded, there is a change occur. The missense means a codon for one amino acid is changed to the codon to another amino acid. Change it to codon for another amino acid. Nonsense. What is nonsense? A codon which is coded, it doesn't go and make over the amino acid, but it is end the nucleotide sequence. That is also called as termination or stop codon. That is called a termination or the stop codon. The fourth one, frame shift. What happened? The frame will be shifted, which confuses the mRNA to read the message from the frame, and there will be a alteration in addition and insertion of the nucleoside pairs. Okay, thus it has a difficult forces. That is also called as frame shift mutation. And children, let me have the next task, that is a mutagen. What is a mutagen? The factors which cause the genetic mutation. The factors which cause the genetic mutation is referred as mutagen. It is also called as mutagenic agents. It is also called as mutagenic agent. Children, it's of 2 types. Physical mutagen. The second one, chemical mutagen. Physical mutagen, chemical mutagen. So Muller in the year 1927 denoted the physical mutagen occurrence in the Drosophila. Okay, Muller in the year 1927. Muller in the year 1927 noted the physical mutagenic condition in Drosophila. See, scientists have used temperature and radiation for that process, and here the temperature play a vital role in the mutation. Raising the temperature also raises the mutation to occur. See, there are some other radiations like X-rays, gamma rays, alpha rays, beta rays, cosmic rays, radioactive isotopes, neutrons, UV rays are also induce the mutation. Children, electromagnetic spectrum, see electromagnetic spectrum which has a wavelength, shorter and the wavelength of rays, and on the basis of the shorter and the longer wavelength, the radiation will be of two types. What are they? Ionizing radiation, non-ionizing radiation. It is more efficient than the visible spectrum. Children, it also affects the mutation. It also causes or induces the mutation. The second one is a chemical mutagen. As we discussed, the chemicals are also inducing the mutation. Here some of the chemicals I have given. See, EMS, MMS, formaldehyde, eosin or manganese salt are also causes chemical mutation. Children, EMS, ethyl methane sulfonate, ethyl methyl sulfonate, methyl methane sulfonate. So they are also inducing the chemical mutation. Children, so on the basis of the mutation, we came to have two more important crops which are cultivated. See, we have to be very familiar with the name M.S. Swaminathan. Who is M.S. Swaminathan? Who is the father of Indian Green Revolution, okay, who is the father of Indian Green Revolution. So what he does many experiments on this hybridization technique, okay, and he used many radiation to perform in the crops to cultivate more of the valued grains. The Sarbati Sonara is a best of it. The Sarbati Sonara is formed from the traditional wheat variety where it has been induced with the gamma rays. More number of gamma rays are induced in the cultivation of this Sarbati Sonara. The Sarbati Sonara is a mutant variety of wheat now readily available in the markets. Okay children, it is not a common variety of wheat. It has been cultivated by inducing it through chemicals, radiation. And the second one, the castor aruna. What is a castor aruna? It is a mutant variety of the castor. Okay children, so it is also discovered by Mr. M.S. Swaminathan. What it does, see, induce the castor to perform more, and see the ripening stages of the fruit have been the ripening stages induced to 120 days, whereas the normal ripening stage is 240 days. Okay children, it has been induced to get fruit from the castor within 120 days by inducing neutrons radiation, Let me continue this in detail. Children, as I said before, the M.S. Swaminathan who have discovered the mutant variety, that is Sarbati Sonara and the castor aruna. Is it not children? We are using it in the commercial way. We are having it in the provisional stock. So these are the varieties are commonly used now and all. Okay children, and the next one is a co-mutagen. What are co-mutagen? The compounds which are not able to have the mutagenic properties, what they does, they enhance the effect. They enhance the effect of a known mutagen and are called what? Co-mutagen. Enhance the effect of the mutagens, and thus there are some co-mutagen. What are the examples? See, ascorbic acid which induce the hydrogen peroxide and make the mutation to occur, and also the caffeine make the toxicity, induce the toxicity of the methotrexate in plants. Okay children, thus the mutation came to occur, which is the co-mutagen which help the mutation to occur. So which is not directly cause the mutation. And the next one is the mustard gas, which also induces dichloroethyl sulfide to induce a mutation. Here the mustard gas was induced. Anyway, it doesn't show the mutational properties. What it does, it enhances the dichloroethyl sulfide to induce a mutation. Likewise, the fruit fly. The fruit fly also do not have the mutational properties. What it does? What year X-rays are induced by the Muller in the year 1928, and thus a fruit fly causes the mutation. Okay, children. And likewise, the Stadler also using X-rays, gamma rays to induce a mutation in plants, and thus Arakkonam also induce the chemicals to mutate in plants. And of course, these are the various types of mutation we see in this chapter. And of course, children, we have discussed about the mutation. What is a mutation? It is sudden change in the genetical setup of an organism is referred as what? Mutation. It was described by Hugo de Vries in the year 1901. And these are the various points regarding the essay of mutation. Thank you, children. We will meet you in the next class by another topic. Thank you, children.